The baseline required for human survival relies on a constant and highly precise regulation of our body's internal environment. At the center of this physiological balancing act is a single, microscopic governor, the hypothalamus. Its control architecture is dual-natured. It simultaneously manages the high-speed electrical impulses of the autonomic nervous system and the slower chemical cascades of the endocrine system. Through these two networks, the hypothalamus dictates our most critical homeostatic parameters. It regulates our sleep cycles, dictates thirst and hunger, manages core body temperature, and triggers systemic stress responses. Look at this 3D model of the brain. When we strip away the outer cortex, we isolate the diencephalon. Here, seated at the absolute base of the brain and directly inferior to the thalamus, we find the hypothalamus. To understand how it functions, we have to look at how it is spatially organized. The internal structure is divided along two intersecting axes. Moving from the inside out, the transverse axis separates the structure into a medial zone and a lateral zone. Longitudinally, the architecture is mapped into three distinct regions from front to back: the anterior, the tuberal, and the posterior blocks. For our current analysis, we will filter out the tuberal and posterior regions. This leaves us with the anterior block, positioned directly above the optic chiasm. Because of this anatomical placement, this isolated anterior structure is formally known as the chiasmatic region. To understand how the brain translates physical environmental stimuli into systemic physical responses, we have to map the exact architecture of this localized anterior cluster. We begin at the anatomical baseline of the anterior region. Resting directly on the optic chiasm, a distinct nuclear cluster forms our first structure. This is the Suprachiasmatic Nucleus, or SCN. Notice its direct spatial proximity, sitting squarely atop the crossing optic nerves. Physiologically, the SCN is the human body's master circadian pacemaker. Cellular clocks naturally experience circadian drift. Without external intervention, our internal timing falls out of sync with the 24-hour day. The SCN requires constant environmental calibration to keep those cellular rhythms accurate. That calibration relies on an environmental input pathway. Ambient light-dark signals are continuously captured by the human retina. Those photic signals are transmitted directly up from the retina, through the optic nerve, and straight into the SCN's architecture. The SCN physically processes these incoming photic signals, using them to synchronize downstream biological clock mechanisms throughout the nervous system. The SCN functions as the definitive biological bridge, physically connecting the planetary rotation of the Earth to human cellular metabolism. Moving superiorly and laterally from the SCN, we encounter the most functionally diverse hub in the anterior region. This is the Paraventricular Nucleus, or PVN. It acts as the primary neuroendocrine command center. Looking at a cross-section of the PVN reveals a critical internal division. It houses two structurally distinct neuronal populations: small, densely packed parvocellular neurons alongside larger magnocellular neurons. The parvocellular neurons have a specific physiological mandate: the synthesis and deployment of releasing hormones. Specifically, this population produces vital chemical messengers like corticotropin-releasing hormone, or CRH, and thyrotropin-releasing hormone, TRH. Once synthesized, these chemical signals exit the parvocellular region, travel down the infundibular stalk, and terminate directly in the capillary beds of the anterior pituitary gland. By delivering these specific releasing hormones, the PVN governs the anterior pituitary, triggering downstream systemic endocrine cascades, like the release of cortisol during stress. But the parvocellular network is only half the structure. The magnocellular neurons operate on a massive physical scale and have a distinct output. These larger neurons synthesize oxytocin and vasopressin, and rather than sending them to the anterior pituitary, the magnocellular pathways bypass that control system entirely. By utilizing this dual cellular architecture, a single localized nucleus can independently orchestrate immediate systemic stress responses, alongside cascading long-term abolic shifts. Neighboring the PVN is a neuroendocrine structure that shares a specific functional pipeline with those magnocellular neurons. This is the Supraoptic Nucleus, or SON. It sits laterally, positioned right above the optic tract. If we contrast its cellular makeup directly against the PVN, we find the SON is composed almost exclusively of magnocellular neurons. Its molecular output is focused on the high-volume synthesis of vasopressin, also known as antidiuretic hormone, and oxytocin. Because these hormones avoid the local bloodstream portal, they require axoplasmic transport. They must physically travel down the actual length of the cellular axon. Mapping this axonal pathway shows it originates at the SON, passes through the infundibular stalk, bypassing the anterior pituitary, and terminates in the posterior pituitary. The posterior pituitary acts as a storage reservoir. It receives the axoplasmic flow and ultimately dumps these hormones directly into the systemic bloodstream to control total body fluid balance. Operating as a manufacturing and biological transit pipeline, the SON completely bypasses glandular intermediaries to exact immediate control over human blood volume. Shifting our focus away from the endocrine export hubs, we look to the nuclei dedicated to intrinsic autonomic computing. The anterior hypothalamic nucleus serves as a parasympathetic control center. Its specific autonomic function revolves around thermoregulation. When the body overheats, it is this nucleus that mediates the necessity of heat dissipation. To cool the body, it triggers physical responses, specifically inducing sweating and cutaneous vasodilation at the surface of the skin. Finally, completing the anterior region, we find the preoptic area. The preoptic area overlaps with the anterior nucleus in monitoring and regulating core body temperature. However, it maintains a unique secondary function, the modulation of reproductive behaviors. To execute this reproductive function, the preoptic area relies on a critical neuroendocrine output, the secretion of gonadotropin-releasing hormone, or GnRH. Both nuclei execute their parasympathetic commands by projecting efferent signaling pathways downward, driving signals out of the hypothalamus and toward the lower brainstem. These two adjacent regions demonstrate that the anterior hypothalamus is as critical for our immediate parasympathetic survival responses as it is for the long-term propagation of the species. Notice the operational architecture packed into this microscopic neurological volume. When we look at the fully illuminated anterior chiasmatic region, all 5 distinct nuclei come into focus. At the base, the SCN constantly processes real-time planetary retinal input. Above it, the PVN and preoptic area simultaneously deploy precise releasing hormones downward into the anterior pituitary. Laterally, the SON actively transports raw fluid balancing hormones down its physical axons directly to the posterior pituitary. And internally, the anterior nucleus fires high-speed parasympathetic signals downward to the brainstem to manage core temperature. Mastering this localized map establishes the basis for clinical neuroendocrinology, bridging the gap between abstract neuroanatomy and the tangible reality of human physiological survival.